As you see, the unraveling the mystery of boron tolerance bacteria. And the approach that will be done, of course, that will be assisted biology approach. Right? Before I enter into the main theme, let me just tell you what is system biology. You should have an idea about what system biology is. Think about an example like if you are asked to describe a forest, what are you going to do? You can say that there are these type of plants that are there. So there are 7,000 or 8,000 sal plants, there's and all the plants you can say, but that is not the description of forest. In addition to that, there are so many interactions that is happening within the forest. So the forest is actually, if you break into smaller units and add up all these knowledges, it will never be a whole number. So always we have done in our mathematics, a whole number is 1 is actually the sum of the fractions. But in system biology, when we're thinking of putting all these things together, it is exceeding 1. So therefore, there is an enormous shift in thinking, in understanding what a system is. A cell is a system, and we practice the art. Yeah, thank you. We practice the art of understanding the bits even 25 years back. Any phenotype linked with a genotype. So we never thought that a small change in a genotype may affect the other phenotypes too. Because at that point of time, the science was growing in such a direction, we were assembling all this information. But today is the point where we are going to handle all the information together. So there will be a huge data that needs to be processed to come to a single important intervention. May I have the next, please? Okay. What is this? All of you know this is Boron. Hatimarka 3. And there is a story behind this. We say that made in India, and that it was done in 1929 by Guru Mohan Datta, GD Pharmaceuticals, who wanted to reverse the entire thing. And this cream contains a very important component called boric acid. So boric acid is being used in this particular cream along with zinc oxide, a sunscreen, and a lanolin, and some perfume in it that make the brand Boron. So when India became independent, 1 lakh such Boron tubes were distributed freely in Kolkata. So that was to mark, coming out of the colonial clutch. But to start with, this boron being used by the human beings for millions of years, thousands of years back, the Egyptians, the Babylonians, they used boron, borax, right? So borax has been used from time immemorial. But interestingly, 1857 was the Sepoy Mutiny. And 1857 is a very, very important day. Why? Because the boron was discovered in plants. So the plant have boron. So there must be some property that is being executed by boron. But interestingly, this boron have a very narrow range of activity. It is required by the living organism, and it is even toxic with that range, that goes out of the range. It is toxic to the plant cell, it is toxic to the human cell, it is toxic to the bacterial cell. So my dear young researchers, we have been dealing with the metal resistance for long, when actually the science started with. But interestingly, no resistance mechanism were discovered that how boron is being resisted by certain bacteria which can grow in presence of boron. So there must be certain mechanism. Like some bacteria can grow in heavy metals. So there must be some reason why it grows. So, but in 2023, we did not have any answer to it. I'll come to that one by one. May I have the next question? So this is boron, a peculiar mineral element, and here you see the atomic number of the mass. And interestingly, this is in between a metal and a non-metal. So it is not a metal, nor a non-metal, it is a metal. Next, please. So why is so special about boron? Because this boron, it percolates in the form of uncharged molecules. Some other heavy metal, they dissociate into ions and they enter into the cell. Interestingly, boron enters the cell in the form of uncharged molecule. And this has a tremendous propensity to enter into a cell which is having a lipid bilayer permeable. So it can readily enter into the cell. And when it enters into the cell, at the level which is not required with the cell, it is going to do a tremendous damage. Right? So another feature is its reactivity with acids and alkaline. This is so special about boron. Next, please. So it is harmful about a certain threshold of boron that I have just told. The extremely narrow range between deficiency and toxicity. The plant scientist, agricultural scientist knows well that how boron deficiency can lead to the destruction of flowers in acres and acres of Jarvera, or sorry, in some other plants also, which is very much. In plants, there will be a boron toxicity. And it is going to affect several developmental and biochemical processes. In lower plants, for example, let's say yeast, what happens? The yeast, if it is subjected to a high degree of boron in its environment, I told you it's going to enter. But yeast has to have a mechanism to pump out these borons out of the cell. So we have found out, certain boron influx pumps in lower plants. Next, please. In animals, also this deficiency is correlated with low immune function. And extraordinary B level will cause a damage. So therefore, you have to have a balance between the deficiency and toxicity, and that balance has to be maintained. Next, please. So what does boron do after entering the cell? This is a very, very important question. So once it enters into the cell, additional metals with well-studied resistance mechanism, they can form complex with other chelators, such as organic acid. But contrarily, this boric acid is a very weak acid, and that functions as a very strong acid when it is inside in presence of specific chemical molecules. Interestingly, when you open up the cell, there will be a lot of glycerols with which boric acid can react and that can produce stable cyclic combination. Borates can form a reversible boric ester complex with intramolecular hydrogen bonding, particularly the nucleoside sugar, which is going to interfere with the base pair interaction. So you can understand that if boron is there in the cell in plenty, it is going to stop all the DNA metabolism. Right? Next, please. So we need to figure out really how, when we talk about the resistant or tolerant bacterial cell, which is really surviving, how it does. So, what he said that you need to have a hypothesis. So, our hypothesis, after going back to all the reference literature, our hypothesis is that a cell can only survive and flourish if all these macromolecules which are damaged because of the entry of this boron, if this macromolecule synthesis are charged up, and that leads to the operation of all the metabolism, then the bacteria is going to survive in presence of high amount of boron. Next, please. So, what will be the approach? I started with the system biology. That means we have a hypothesis. And if we have a hypothesis, then we will have to do a certain experimental work. And from this experimental work, we will have a lot of data. And that data needs to be integrated. And finally, we will make a model of the field. And that's what the system biology is. We are looking at the system from all the things, from gene to phenome. It's the sum total of the phenotype. It's the last word that we look into. Is the phenome. And in between, there will be a lot of regulation. And interestingly, let me tell you that it never correlates. When you speak about the abundance of certain transcripts, which transcripts are actually the product of the genes in the form of mRNA, and the number of proteins that will be ultimately manifested, there is no correlation. Or very low correlation. And this has been haunting us till 2022. Then a path-breaking paper came out in Science, I will come to that, which has taken into consideration about 1,500 the genes that are expressed within a system at a point of time. But interestingly, this 1,500 genes are not yielding protein at the same rate one should do. Then what is the reason? Okay, next please. So one of the significant objectives, as I said, is to understand the regulation of the cell behavior through interaction of various components of radiolome. How is that we talk about OMICS? So I told you in the title that it is OMICS. So there are a lot of OMICS. So when we talk about the genes in totality, that is genomics, that's genome. When we talk about RNA getting expressed, hundreds, thousands of RNA being expressed within a cell at a particular time. When you are studying that, that is called transcriptome. And out of this transcriptome, some proteins are really getting fixed in the cell. And when you study that, that is proteome. So all these omics, right? When you put together into a regulatory network, we call it as regulome. Next, please. So everyone has a startup. So come at the moment, come at the man. So I dedicate this lecture to a person who is no more, happens to be my mentor, Prof. And this Prof. was studying this bugs actually, which is purely inorganic chemical processes by which it survived. And I happened to work with the chemoautotropic organism, and I was sitting in the oxygen monitor for several hours trying to understand the physiology of the cells. At that point of time in 1989-90, I understood the vacancy and the void of our knowledge. Right? So the journey started from 1990, and this is the gentleman who put me into this question long back. Next week. So what is it? Keep asking yourself. This is a great statement of a mentor. Keep asking yourself whether in this world, brimming with microbial genera and genes, can't a single one be mind the stone? Actually, it's talking about a pathway. So we were working on this sulfur oxidation pathway, and eventually within a decade we entered into discovering a pathway. The source of Boron pathway was actually discovered from a molecule. And their life processes were played out in a very simple fashion. And he used to play with these parts, and we are, of course, were playing with that. Next. So I already talked about the biological effects of boron that can affect animal, plant, human beings, and the microbes. So the best way to understand really how boron can be tolerated, even by the plants or the animals, you have to have a model that is a bacteria to understand. So till then, we did not understand really how bacteria is capable of resisting or tolerating boron. For other minerals, heavy metals, everything discovered. And that happened in before 2000. We have to wait till 2023 to really unreveal the answer that how boron is tolerated. Next, please. So this is already been spoken, and because of its inhibitory effect on microorganisms is also affected. So we have to start up with something from where we are going to get this organism which can tolerate boron to a big extent. And the answer was the agricultural fields. For cauliflower vegetation, one of the most important things the farmers do is to add borax in the field. If you don't add borax in the field, your cauliflower production will come down. Right? So definitely we start wanting to understand that the microbes there in the field must be tolerant to boron because a huge amount of boron is used in those agricultural fields. Next, please. So bacteria that grow in high boron environment, a lot of references before we started working on this. And these are all the different bacteria which were reported to be boron tolerant. And the tolerance level is 450 millimolar to 60 millimolar, 300 millimolar, and the list is on, and these are the references. But unfortunately, none could find out the reason of intolerance. Why? I'll come to that answer. Next, please. So the list goes on. Next, please. So different types of, so if it is happening in the bacteria, there must be happening in the plant and the smaller plants too. For example, as I told you about the yeast. So the yeast, they have found out certain transporters which are responsible for tolerance. Right? That means, I told you already, if there is incessant flow of boric acid inside the cell, there has to have certain mechanism to push them out. So there must be some influx. Should be there in also in case of bacteria. Unfortunately, nothing, no influx transporter would be just, we can point it out that this is responsible. Next, please. So how is it was designed? So designing is very, very important. When you experimentally design, how it was designed, you get the boron tolerant bacteria from bone-rich soil, and then you standardize the methods to ascertain the tolerance of those. Then you apply in vivo engineering principle. This is very, very interesting. What do you mean by in vivo engineering? Now, what you have heard already is you can engineer a strain, you can engineer a gene, you can express some genes in a bigger ways to get the phenotype. But if you allow the cell to remain under stress condition for several months of years, you need not to do recombinant DNA. The cell itself will engineer to cope up with the additional stress that you are applying for a huge amount of days. And to a bacteria, To us, a year is 365. But to a bacteria like E. coli, every 20 minutes it grows. So several hundred thousands of divisional cycle will take place, and that is going to allow the engineering form within to come up with a strain which can be adapted to a much higher boron that in our case we were doing. Then to reveal the molecular basis of boron tolerance. How could these cells really tolerate boron to an extent of 200 millimolar, and which is typically a very high amount of boron that were tolerated by our strains? Then understand the effect of boron on soil bacterial diversity. So the knowledge that is being revealed that has gone to the field. So if you have seen a boron infected soil, it is perfectly desert, right? No plant even grow there. So it is boron induced desertification, right? We'll come to that at the last. Next, please. So isolation was an important even that we need to have our own system where we will have to work. Next. So this was our sampling site. I already told you that it will be a polyglot planting station from where we collected the soil sample. Next. And if you look at these sampling stations and we look at the boron concentration of those soils, that will give you an idea, even if you do not, you just look at this, 90, 60, 50, 45, and the boron toxicity arises when the soil has more than 12 ppm. So more than 12 ppm is there several times of 12 in these soils. So we thought, these soils would be the best to find out some bacteria which can tolerate and grow. Next, please. So you see, this is the critical enrichment technique a microbiologist do. We take the soil and with increasing boron concentration, you go on giving into the next, and finally, at a point of time, we need to isolate those. Because by this time, the bacteria which are capable of tolerating a high concentration of boron has already been stabilized in the community, and you end up with the isolation of boron resistant or tolerant. Next, please. So they were cultivable boron tolerant bacteria, and they were named accordingly as per the soil Ma2, Ma3, or whatever. These are the different soil samples that have been worked out and that gave us in successful isolation of bacteria. Next. So when we are determining the upper limit of boron tolerance, so how great this OL1 can tolerate? You see, it's moving up to 250. So different strains that we're isolating, purified, we want to have an understanding about the upper limit. So we will be working with these host organisms which have a very upper limit of tolerance. So you can see here, this OL1 has a big tolerance level. Next, please. And when you group these isolates on polymorphism of restricted 16S RRN and gene, so all these isolates, from there you take the 16S, digest with a rare cutter, with the same rare cutter, and if you run, there is a polymorphism, which will lead you to understand that the strains belong to the same group or a different group. Next, please. And these are all the acceleration number of the strains that has been isolated and which has a 16S RNA and gene similarity with the closely related species. Next, please. And finally, we wanted to come here. This OL1, this gives you a phylogenetic tree of OL1, OL2, OL3, OL4, and OL7 based on the 16S RNA gene. Next, please. And this OL5 and OL6 was belonging to another genus called Enterococcus. And you end up with a two different Enterococcus species, OL5 and OL6, which is different from its nearest one, Enterococcus acidophilus. Next, please. So these are all the bacillus that were isolated, and the nearest one is the Bacillus anensis, to which we form the cluster. Next. Next is the drought genome. It was very important because we want to ascertain the gene expression in these bacteria. So the publication resulted with the drought genome sequence of two boron tolerant, arsenic resistant, gram-positive bacterial strain, OL1 and OL5. Next, please. So when you this is nothing to know, these are the colony morphological study, you find a difference in its characteristics. This gives that difference that OL1 and OL5 perfectly dissimilar in morphology. Next, please. And when you look at this scanning electron microscope of OL1, terrifically big, bacillus, you can see the picture, and you can see OL5 is well like a coccus, and we told that this is the enterococcus, and the other one is lysine. Next, please. And these are the biochemical study results that when we compare between these two strains in biochemistry, they have a very less similarity. The index is 24.44%. Next, please. And when you are trying to optimize the growth parameter, you find that so OL1, they love the particular temperature, the nutrient condition, NaCl condition, and the pH. These are needed to be studied when you are really planning for a transcriptome study. Next, please. So then we try to find out is there any relationship with boron resistance with the other metal resistance like arsenite, cadmium, chromium. This is another metalloid. Copper, mercury, zinc, nickel. We found really they are resistant to some others. Right? Next. So when we tried that, we had in our mind that if these isolates really being a useful isolate in the boron field, it should have certain plant growth promoting activity too. So these were studied, the indole-acetic acid production ability, the ability to solubilize phosphate. These are very important agricultural traits. Right? Next. And these are the different states and biofilm formation. Everything was studied between these two strains, OL1 and OL5. Next is, so what is the key finding? The key finding is because we split this highly boron fortified soil we can use, and for the first time the diversity in culturable boron tolerant bacteria is explored. And this is going to open new door to uncover molecular mechanism of boron tolerance. So we have to standardize what is happening within the cell. So when it is being subjected to a high amount of boron, whether the cell is retaining the boron inside or pushing it outside, how much it is retaining within the system. So you need to quantify boron within the cell membrane, and also externally. Right? Next. So we designed the physiological experiments to go into the details at the cellular level. So when you start doing regulation, you have to understand whether you can induce this resistance or not. Go back to your basics where we study the induction. Induction is a very small amount of the metal or that nutrient. We charge up the expression of all the genes that will be required to get it tolerated, boron tolerated. So, extracellular, intracellular estimation of both these strains, and they were done, and the estimations. Next, please. Then we use the cell for sonicate. You have to sonicate the cell to understand the boron quantity in the cell life cell, then the intracellular boron concentration in boron pre-exposed cells. If it is exposed to boron to a very small extent, what is their mutation? All these detailed physiological studies were done in order to have an idea about the regulation. Next, please. So these are the way you calculate the minimum inhibitory concentration of PA beta N. So by this time we came to an understanding that boron is to some extent influxed out. So if it is influxed out, then if you try some influx inhibitor, then what will be the level type? So that's why we started using PA beta N against these two and try to understand the tolerance levels have shifted or not. So if you stop influx, then what's going to happen? It is going to be a little bit sensitive, isn't it? So let's see what happens. Next slide. So these are the unraveling of the physiology under boron stress, where we worked out the induction concentration, and you see there is a progressive reduction of the lag phase, and it is entering into the log phase which is pre-compute. So these are the classical regulatory physiological experiments that we have been performing with every disease. Next, please. So, inducibility of the nature of the boron resistance was established, and we knew that boron resistance is inducible. Next, please. And if it is the influx, and we try to measure the extracellular, loosely bound, tightly bound, intracellular, very interesting. If you look at this figure, you find intracellular boron concentration is much less below the 20 millimolar or 0.8 millimolar, 10 millimolar boron concentration, where in the external, there's always 200 millimolar. So what's happening? It's pushing out. So, next slide, please. So these are all the picomole per microgram. So you have to have this quantification to a very great. Next, please. You see, when we have used PA beta N, surprisingly, what we thought that it will be completely sensitive. No, it didn't happen. It partially it came out from 230 to 150 and 160. So when you're using influx inhibitor, it became sensitive, but not totally sensitive. So partially these PA beta N acting on those influx inhibitor, influx transporter was active. That has made to this reduction. Next, please. So the key findings is the boron resistance capabilities of this new microbial isolate. That is the starting point to understand and validate our hypothesis. Next, please. So, putting all these physiological data, the first breakthrough paper in understanding the physiology of boron resistance came out in Biometals and Biotechnology. Next, please. Now it is the time to enter into the revelation of molecular basis of boron tolerance. So we have to use a suitable bacterial strain. We cannot go with the two. So we settle with the lysine bacillus. Oil one. Next, please. So what is the baseline correction? As I said, that is the central dogma. It was generally assumed that there exists a direct correspondence. What is that correspondence? The explanation of the environment transcripts and the generated proteins. But it does not happen. Why? The correlation between mRNA and protein expression can be low. And the answer I will give you later. And that is due to various factors. the half-life of the mRNA, and think about the situation. Think intelligently. Those who are hearing this lecture, try to find out or speculate the situation in a state, there are 1,000 mRNA. And there are only some fixed number of ribosomes. So ribosomes should have access to all the mRNA at a particular rate. So there are transcripts to which the RNA, the ribosome binds, there is a splicing. There is a change, and the abilities are varying. So therefore, the net expression which you get in form of protein that will come out with the proteome experiments, you see it later. So this is the baseline connection. So let's move to the next. So in order to do this, you have to have the whole genome, because until and unless you have the whole genome, you don't know really which are the genes, and you have to have the annotation of the genes. So we have both the OM1 and OM5 sequenced in two different platforms. One is Illumina and another is Android, and ultimately we have all this genomic analysis. So that's the part of genomics. So we complete the genomics. Next, please. Then, with the use of the whole genome sequence, we try to relate and understand where this particular your isolate belongs to. And then we select the suitable boron tolerant isolate, as I told you, that the OL1 will be our model. And next, please. Let's read the subject of the transcription. So this is the experimental flow. So day one, you isolate the RNA and construct the cDNA, goes through the sequencing library preparation. Day two, you have an emulsion PCR, then you enrich, sequence, and finally you assemble at the data process, and you annotate and map the transcript quantification. That how many transcript per chain has evolved. So it's a huge genome, right? Several thousand gene expression you start understanding at one go. So this is big data. Next. So this is the total RNA isolation of these two. And this is UC fast. Next, please. So total mRNA enriched mRNA reverse that I have just shown. So next. And then you map it on the reference gene. Already we have the genome, so now you start mapping it on the reference genome, taking the raw reads, take the high quality reads, you map it on the reference genome, then you assemble, and then you find the dates. That is very important. Differentially expressing genes under boron condition and under normal condition. Next, please. So then, after the transcriptome, you have to do the proteome. Otherwise, nobody will accept that even after the transcript abundance will not speak about what really happening inside. So we did the proteome expression of these. Next. And you know these are protocols, standard protocols by which we develop, we got all the proteins that express under boron influence and not under boron influence. They needed to be compared. And again, you have to have those data of differentially expressed proteins. So you have a differentially expressed transcripts, differentially expressed proteins. Now you correlate these two data. Next, please. So the heat map must be constructed, and after that we will enter into the deep area of protein-protein interaction analysis. That what is really happening within the OL cell. Next, we have to put and form the clusters, and then from the cluster and module analysis to find out the hub genes. What is hub? You have seen a cycle wheel to which the spokes are attached. The axle moves on that. That central region is the hub. So we need to enter into the hub and understand really how it is regulated. I told you that we have to understand the main law. Next please. So genomic features of these two will not be so much important for us. Next please. So this is the phylogenetic analysis of the whole genome with 49 genes being used, and even here we find out that lysine bacillus one stands separate from any of the lysine bacillus strains yet discovered. This same is true for Heterococcus species worldwide, and I told you that we use 49 core universal genes defined by the core. Next, please. So genome comparison, it gives a comparison of these two strains. Next. And lysine bacillus capsaicin is very much near to it. You find these values. DNA hybridization value and ortho value. This gives you an idea about how close these two strains are. Next, please. Next. So this is the Heterococcus cassiopeus, which is most near to the worldwide. Next. Now, what is the result of the whole genome? This is a very routine thing when you have all the genes. Now you just divide into subsystems, and this is the subsystem which it counts, and this gives you the graphical representation of the total genes that is present in the bacillus strain OL1. Next, please. This is for worldwide. Push on. Next. Now, is the question of genome mining. So the part that is being played by the software is over. It is a part of the human brain to analyze. The equals is an important thing. Then you have to find out, you have to mine out these equals proteins in the genome of well. So that we did. Next, please. Then the PGPR phenotype is given, so there must be a genotype related to it. So we mine those genes that are related to PGPR genes. Next, please. Now, the transcript. T means control, T means test. Test means in presence of boron. In the presence of boric acid. You find the number of E reads, total number of base, tremendous amount of data in terms of GB. We have attended 2.95 and 2.6 GB data of control and T, and now really the real work of the bookwork starts. Next, please. So when you distribute them, it's very interesting that most of the expressed genes are annotated. They are found to be associated with amino acid metabolism. Interesting, I told you when boron enters, it actually takes over. The entire protein synthetic machinery is down. So the cell has to increase the protein synthetic machinery to yield proteins. And if those proteins are not produced, you will not have the supply of ADP. So what's happening to those metabolisms, which actually involve in producing ATP, in generating ATP? So the thing is, the energy supply line must also be upcharged. So we were getting more and more passionate and going towards really what we wanted to have. Next, please. So if you find the enriched pathways in response to the boron, interestingly, you can also answer that glycolysis, nitrate, they are involved in giving you this energy support. And number of dates means number of differentially expressed genes. Here you find, and tremendously you will find in translation. Translation is the protein. Proteins are getting destroyed in presence of boric acid. So more number of proteins have to be produced in order to titrate the damage. Same is true for the influx associated. 39 tags are very full. So translation, influx associated stress, and amino acid metabolism all show the rate tax. Next. And if you look at the heat map analysis in presence of boron and in absence of boron, this redness is the highly expressed genes of carbohydrate metabolism placed as a heat map. Next, please. This is efflux transporters. You will see that in presence, in absence of boron and in presence of boron, is a striking change in the expression of efflux-related proteins. Next, please. When you look at the stress, because already that is stress, so it has to mitigate that stress. So what about the mitigating? You see the large panel of red genes. These are the expression of the genes that are being expressed in presence of boron. Next, please. This is genetic information processing. The cell has to survive because we transmit the DNA from its to the survivor cell. So genetic information processing must go properly. And you see what happens in the absence of boron. These are the proteins upon which we are going to work to find out the hub genes. Next, please. This is the nucleotide metabolism. It's also you find a slight difference in presence and absence of the gene of boron. Next, please. This is amino acid metabolism. There are certain enzymes related to production of certain amino acids. They are highly expressed in presence of boron. Next, please. Now we will have to enter into the proteome to make our understanding clear that whatever the transcript abundance that we have observed and analyzed is really reflecting the proteome. What I choose to do? 230 dot definition mentioned. My age story. So pathway enrichment and heat map analysis which enable us to corroborate the transcript data or transcriptome data with proteome data. Next, please. So you see in presence of boron and in absence of boron. Oh my God. In protein expression, transcription and translation are the rate ones, replication and repair more or less the same because it has to replicate and repair. So this is the heat map which is created on the basis of total number of proteins per pathway. Next, please. So again, transcription, translation, stress and amino acid metabolism. The dates that were expressed in transcript and the proteins that are expressed in proteome. Talks. Next, please. So this is what heartbreaking word expressed in science where they did the maths on central dogma. And I was telling you that story. You have to have an abundance of more than 1500 genes in the bacterium to understand the correlation between the transcript abundance and protein content. This is the first biggest attempt to understand and correlate proteome with transcriptome. Next, please. So they did the mass on the central, and they these are all will be useful for design of synthetic circuits in bacteria, and we'll do that. Next, please. So the string database giving you a prediction of several hundred protein in a mess. This is the protein-protein interaction network, which were produced with significantly upregulated takes in OL1. Next, please. The proteome data also gave you certain proteins that were expressed, all put together. Next, please. You break up the clusters. That big cluster was broken down. We got 6 densely interactive clusters in that PD1 in which these proteins were involved in various biological pathways. And we did this cluster with all the calculated tags using encode. Next, please. So this is about the encode, what were the cutoff score, which he said that you have to be very alert. You can run something, you can get the data by default, but that does not really allow you to come to a meaningful information. Next, please. So this cluster one has shown proteins, very interesting, that were involved in the RNA metabolic process, tRNA binding, translation, transcription, and initiation. Next, please. And this is the constructed pathway network of the cluster 2, and which was involved in the nucleotide binding process. 2 cluster 3 were involved in butanoid and purine metabolism and TCA cycle. Next, please. And this is the cluster that were involved in purine, nucleoside, monophosphate biosynthetic process. These are the constructed pathways network from cluster 4. Next, please. This is the cluster 5 and cluster 6. They were participating in the cytosolic process, the carbohydrate derivative binding, small molecule binding, small molecule metabolic process, etc. Next, please. Now the very important conclusion. When you have all this data, then we have to end up with the hub gene. Interesting to note that we have identified 10 hub genes. And these are the genes which are involved in ribosome biogenesis. Next, please. Very interesting hub gene is RPLNP. What is that? It codes for the binding of the protein. It codes for the protein that binds with the L16 of the nuclease subunit. And this binding is required for the stimulation of gene phase. And that is required for the release of RPGA from the ribosome. And if that doesn't happen, the conversion of the intermediate to complete TS will be halted. And this hub gene was predicted by 9 algorithms. Right? And then RPSD, which codes for S4, RPL4, and this is predicted by 8 algorithms, and 2 more hub genes, RPLE and RPLA, were predicted by 7 algorithms. Next, please. So, finally, bacteria will be needing a lot of energy to thrive in this unfavorable environment. Protein synthesis and amino acid metabolism are hyperactivated in order to produce the enzymes that are necessary for metabolizing activities. Right? So what should be? Next one. This is the model. So when there is a boron, these are all upregulated. The replication, transcription of different types of RNA, the translation, responses are ATP dependent transporters are up. So gene regulation of certain metabolism must be short-term in order to supply the ATP to conduct the entire experiment. Next, please. This is what we published systematically of 2 months ago in molecular omics, what transcriptomics and proteomics can tell about a high Boron pattern, Boron tolerant bacilli, the viewers, next please. So convinced they wanted the artwork of that particular journal. Baba came up our idea, the fire inside, and all these are actually the abstracts of all that I have been given you. Next, these are the contributors to the Boron story. Subodit was leading the experiments, still the metagenome. And Sridhara was doing all the bioinformatics analysis, the network analysis. Tilok was there in that part where we are working with Genomics, please. He is from Bosch Institute, Shobhushati Bhattacharya. Nidhi was a master's student. Vivendu worked under me, and now he is in the Bosch Institute. Senior researcher completed. Next, So from Boron Desert, whole desert of Ladakh, to the deepest of the Arabian Sea, I had an opportunity to contribute only because of my trusted friends. Next, please. These are all my trusted friends. He is Vidhiman Ghosh, who works hand in hand in ocean to Ladakh to Boron. And this is the group that is involved in sequencing. And this is the guy who works in the Indian Ocean. He's from NIO of the University of Sydney. Thank you very much.